Now that we've gone through the process of DNA replication and all of the enzymes that are involved in that, we want to talk about errors during DNA synthesis. Now, it's really important that the DNA is copied perfectly every single time we do go through a round of interphase followed by mitosis and cytokinesis. There are many mechanisms that are in place to help ensure that the DNA is copied perfectly. But regardless, there are going to be a few errors that do squeak by, that we do have end up in the new DNA that's being synthesized, and then it will obviously end up in the cells that are going to be produced by mitosis and cytokinesis. If we look at the error rate is 1 base pair per billion synthesized. So 1 base per billion. If you consider that human DNA has about 3 billion bases, then we're getting about 3 errors in every round of DNA replication. Now, one of the reasons why the error rate is so low is because the DNA polymerase not only is reading the old DNA and making new DNA from it, but it is also proofreading any DNA that it makes. So what this means is that the DNA polymerase is looking at the old DNA template. If it sees, for instance, an adenine or an A nucleotide there, it knows that it needs to incorporate a thymine across from that. So the DNA polymerase is going to pick up a thymine nucleotide, it's going to attach it to the A, and then it's going to look at what it's done. So it's going to check and make sure that we are getting the correct hydrogen bonding between the T and the A. So we want to get 2 hydrogen bonds there. And it's also going to check for that consistent width to the DNA, keeping in mind that we're supposed to have a purine base across from a pyrimidine base. Even though the DNA polymerase is double checking, it will miss some of its errors. If it does see an error, it's going to back up and remove it and try again. We do have a couple different mechanisms in place to replace any errors that squeak by. So one of those is going to be called mismatch repair of DNA. In mismatch repair of DNA, we're talking about one base that happens to be incorrect. And in that case, we have specific enzymes that are going to be scanning through the DNA, and they're going to be detecting that. So for example, if we happen to have an A that was across from a C nucleotide, if that was the case, it is an A that is a purine and it is paired with a pyrimidine, which is the C, but the problem there is that the A and the C are not going to be hydrogen bonding with each other. So we'll have absolutely no hydrogen bonding between them. So that would be detectable by enzymes that were looking for those types of errors. Just want to emphasize again that we have a consistent width down the DNA molecule, because we always have a purine across from a pyrimidine in all of our base pair combinations. And we also have 3 hydrogen bonds that are going to form between every G and C, and 2 hydrogen bonds between every A and T. If we happen to get any of the bases in a different combination, there will not be any hydrogen bonding that takes place between them. So that would be something that could be detected by repair enzymes. Now, mismatch repair is going to be taking place quite a lot during, say, G2 of interphase, which is when the DNA is being double checked. Those sister chromatids are being made sure that they are exactly identical to each other. Now, DNA replication is not the only time when errors can happen in the DNA. So all throughout our lives, every day, our cells are exposed to a number of things in the environment, and those things many times can cause changes or damage to the DNA. If you think back to the end of chapter 12, we had a number of things that were considered carcinogens or cancer-causing agents. Different chemicals, viruses, bacteria, things in the environment, radiation. All of those things were considered carcinogens because they had the potential of changing the DNA. So if we do get changes in the DNA, anytime during the cell cycle, nucleotide excision repair is around to fix that for us. In nucleotide excision repair, we're talking about larger areas that are damaged. So with the mismatch repair, it was 1 incorrect nucleotide. Now we're talking about maybe 2 to 10 bases that are incorrect. In the example that you see on this slide, we have what is typically called a thymine dimer. Now it's called a thymine dimer because basically we have 2 T's that are across from each other right here, are next to each other, and they're across from two A's, which would be typical in DNA. T's are always supposed to be across from A's. Well, what happens here is that when we have exposure to UV, ultraviolet light, many times it causes two thymines that are next to each other to hydrogen bond to each other rather than the A's that they are across from. So what we end up with is this little buckled out area in the DNA. This makes that region of the DNA very difficult for the cell to read or utilize. So we need to find that damage and we need to cut it out, remove it, and replace it with some good thymines that are going to hydrogen bond to the A's that they're across from. The enzyme that's going to actually find the damage and cut it out is called nuclease. So nuclease, this new enzyme is going to remove the damaged DNA. When it does that, it's going to not only remove the two thymines, but it's also going to take a little bit extra on either side. And notice that we are not messing with the strand of DNA that had the A's or that was not damaged. The reason being is we're going to use that strand as a template to replace what we just removed. After the nuclease enzyme removes the damaged DNA, DNA polymerase is going to come in, and DNA polymerase is going to make DNA. So it's going to make DNA in the same manner that it did during DNA replication. It is adding to a 3' end. In this case, the 3' end is an end that was already present in the DNA, so we don't need to have a primer in this case. We're just going to add directly onto the available 3' end of the old DNA that was there, and we're going to run right up to where the 5' end of the old DNA begins. And then just as we had with DNA replication, the DNA polymerase is not able to make that very last phosphodiester bond. So DNA ligase, same enzyme we had before, is going to be coming in and it is going to join the pieces together. So in this case, we have removed the damaged DNA and we've replaced it with some good DNA again. Now, nucleotide excision repair is going to be taking place every moment of every day, pretty much in all of our cells, because again, we're constantly being exposed to things that could cause damage to our DNA. The nucleotide excision repair does a pretty good job of fixing any damaged areas, but there is a chance that some of the damage will squeak by and it will not be caught and detected and fixed. So that's one of the reasons why we always say minimize any exposure to things that we know cause damage or mutations to the DNA, because we can't be 100% sure that the mechanisms inside of our cells are going to see the damage and fix the damage. So we definitely want to minimize it, because depending on the random area that is affected by those carcinogens, it could lead to cancer or some other types of problems inside of our cells. Another thing we want to talk about with regard to DNA replication and repair is that because we're dealing with linear chromosomes in eukaryotic cells, every time the DNA is replicated or copied, the ends of the eukaryotic chromosomes are going to get a little bit shorter. And those ends of the chromosomes are called telomeres. So what we're saying is that the telomeres are shrinking every time we copy the DNA. And we do copy the DNA quite a lot, because if you consider, we all started as one fertilized egg. That zygote at that point divided by mitosis and cytokinesis many, many, many times to give rise to all of the cells that now make up our body. And additionally, those cells are still undergoing mitosis and cytokinesis for tissue renewal, growth and development, and so on. So if we allow the chromosomes to shrink with every round of DNA replication, that could present quite a big problem for the cell. If we look at why they're shrinking, first off, here as we see in this image, this would be the end of a chromosome. So keep in mind that our origins of replication and the replication forks and things that we looked at earlier in the chapter, they were inside the chromosome, scattered about, but those replication bubbles were expanding as the leading strand continued, and then the lagging strand kept backing up and adding a primer and extending it. Well, eventually we will reach the end of the chromosome. When that happens, for the leading strand, which is shown as the top strand here, that's not a problem. The DNA polymerase continues to synthesize until it gets to the very last base, and it puts that one in. Now on the bottom strand, which would be the lagging strand in this case, if we zoom in on it here, we would have an RNA primer that is added by primase right on the very tip of the old DNA, and then our DNA is going to get extended in this direction, and that DNA is shown as the light blue in this picture. That DNA polymerase would remove this previous RNA primer, fill in underneath, and then ligase is going to seal that gap together. Well, we can remove the primer that was on the end right here, but the problem is, how do we fill in that area with new DNA? Because the DNA polymerase is very much incapable of adding on to a 5' end of DNA. And we don't have anywhere else that we can add a primer to extend and fill in that gap. So if this continues, every single time we go through a round of DNA replication, we're going to be missing a little bit on the end of the lagging strand. So we're not going to be getting that copied. There has to then be a mechanism in place that is going to help prevent this from happening. Because if our chromosomes are shrinking, we're losing valuable information that our cells may need to make some vital proteins that they cannot survive without. So the enzyme that is involved in this is called telomerase. So telomerase basically extends the telomeres. So because it extends the telomeres, we're not going to have such a problem with this. So if we look at this image, what telomerase is going to do is it's essentially going to add on some extra DNA on the end here, which essentially moves the 3' end out quite a lot. If we get that 3' end extended with DNA, and now it's a little bit longer, that then gives us a new place to add a primer. So this would be my primer that I'm adding on here, and that primer would be added on with primase just as it was before. And now the DNA polymerase can come in and it can fill this in, and then ligase can come and it can seal that gap together. Eventually, this primer will get removed, so we won't have that on there anymore. And we will still have a little bit of a gap at the end, which we have not filled in with DNA. But the part that we are now missing is just extra sequence that we added on to the end. So every few rounds of DNA replication, telomerase enzyme is going to come in, it's going to extend the ends of the chromosome, and it's really just extending them with some random DNA sequence, because the purpose of that is just really to provide a spot to place the primer so that we can fill in the DNA that we actually need. So this area right here is the DNA that we really don't want to lose. And this way we make sure that when the chromosomes do shrink, because they will shrink because of the fact that DNA polymerase cannot extend a 5' end, when they do shrink, we're not losing the important vital information in the chromosomes. All of our cells have very good working telomerase to start off with, but there is a lot of research that is currently going into the telomerase enzyme, and it is believed that telomerase plays quite a role in the aging process. So as we get older, telomerase does not appear to work quite as well, and it is not as active inside of our cells, which means that then the chromosomes do start shrinking a little bit and losing some of that information on the inside. Now that's all the information for DNA replication and repair that we have here in Chapter 16. The last little bit here really ought to be a review for us from Chapter 12. And that's just going over the organization of the DNA in the nucleus. We want to remind ourselves that the DNA, although it is a double helix and it's a very long, slender molecule, it is going to be wrapped up and organized around a lot of proteins inside the nucleus. Those proteins are specifically called histones. So histones are proteins that help to organize the chromosomes or organize the DNA. Those histones are organized into kind of beads or larger structures, and those larger structures are called nucleosomes. So a nucleosome basically contains 8 histones organized together into a little bead or a spool, and we're going to wrap that DNA around the nucleosome in a very organized fashion. So they are wrapped around, the DNA is wrapped around the nucleosomes 2.4 times, and then we have a specific number of bases in the DNA that spans between the nucleosomes. And then we also have special ones, histones. This one's called H1, and this is like a linker that is going to help bring together those beads. So what we're doing is we're coiling up the DNA and we're wrapping it very tightly around these proteins, which is going to organize it, prevent tangling of it, and eventually, if we highly compact the DNA, then we get to that short, fat, X-shaped chromosome, which is what we were looking at during the mitosis process. Now keep in mind that most of the time, the DNA is not going to be as highly compacted as what we saw during prophase, and it's not going to be completely naked DNA or DNA that's not wrapped around any proteins. It's going to be somewhere between the two extremes. Some areas of the DNA will be more compacted than other areas of the DNA, and it really does all have to do with how often that particular region of the DNA is going to be accessed by the cell. So if it is a region of the DNA that contains information for making things that the cell needs on a daily basis, that would be a region of the DNA that's going to be more unwound or be less wrapped around proteins. And if it happens to be a region that we use on rare occasion, then that would be an area that's going to be more wound up inside the cell. So make sure we do understand what chromatin is. Chromatin is the DNA wrapped up with some proteins. And then if we're talking about one whole piece of chromatin, that then is what we call a chromosome.